Hi guys, so today we're going to be reviewing Chapter 10, which is all about molecular biology and DNA replication. So to start off, we're going to be talking about the structure of genetic material, and DNA is the genetic material. And this all started off with Griffith's experiments that basically explained how bacteria were capable of transferring genetic information through the process of transformation, and he basically proved this by using mice. And what he did was, say he had one mice and he would inject it with a rough strain that was non-virulent, this mouse would live. But then if he were to inject another mice with a smooth strain that was virulent, the mouse would die. And by doing this, he was able to prove that the genetic material of the virus would be passed down to the mice, and it would ultimately combine with the DNA and genetic material of the mice, leading it to die. Now, in the Hershey and Chase experiment, it basically concluded that DNA, not protein, was the genetic material. And from this experiment, it could be determined that there was a protective protein coat that was formed around the bacteriophage, but that the DNA, the actual internal DNA, is what was passed on to produce progeny inside of the actual bacterium. So DNA and RNA are polymers of nucleotides. So basically a nucleotide is just going to be one singular nucleotide, which consists of a sugar with a phosphate group and your base pairs. And then a polynucleotide is going to be a group of nucleotides. So DNA and RNA are polynucleotides because they consist of more than one. The baseline rule is that it's considered a polynucleotide if it has like 13 or more nucleotides, and DNA and RNA clearly have a lot more than that, so that's why they're considered poly. DNA is deoxyribonucleic acid, and it's double-stranded, whereas RNA, which is ribonucleic acid, is single-stranded. And an easy way to remember the difference is that they both contain different sugars. So purines versus pyrimidines goes into the base pairing of DNA. So purines are going to be your bases of adenine and guanine, and pyrimidines are the bases of cytosine, thymine, and uracil. So here we're going to be talking about the actual structure of DNA, and it was concluded that DNA is a double-stranded helix, and this was affirmed by the experiments of Watson and Crick, as well as Rosalind Franklin, who actually discovered this first, but then everything was verified, and most of the credit is often given to Watson and Crick. So what we mean by a double-stranded helix is that there's essentially a ladder of base pairs that forms in this strand of DNA, and it's twisted. So in order for a replication to take place, this helix has to become untwisted so that we can actually gain access to the bases. Now, Chargaff's rule is pretty simple. It basically states that the amount of adenine has to be equivalent to the amount of guanine because the amount of adenine has to be equivalent to the amount of thymine because these will ultimately pair together because one can't exist on its own without the other. So say I have like 50% of adenine, I have to have 50% of thymine as well because they're going to base pair together, and the same goes for cytosine and guanine. Now, covalent bonds occur whenever there is like an electron being shared between two different molecules, whereas hydrogen bonds are weaker, and these hydrogen bonds are the bonds that forms between base pairs. So as we can see here, the bond between adenine and thymine is a hydrogen bond, and the bond between cytosine and guanine is also a hydrogen bond. So you're going to have 3 hydrogen bonds between guanine and cytosine and 2 between adenine and thymine. Now, in DNA replication, it's a semi-conservative process, and we can see that here. So basically, in DNA replication, since we unwinded the double helix to access the base pairs, this now ladder is going to come apart so that we can start adding new bases. Now, even though when we look at this picture, it looks like a very simple process, it's actually very complicated, and this is due to the number of enzymes that are involved because this process process can't be facilitated on its own. You need things like DNA ligase and helicase and DNA polymerase to kind of get the action started and also conclude it as well. Now, this is a very fast process, so what this means is that there's a great likelihood of mutation taking place, because with this happening so quickly, it's very easy for there to be a mishap somewhere, and this can ultimately lead to problems down the road. Now, DNA replication proceeds in two directions at many sites simultaneously. So 3 enzymes that we're going to take a look at right now are helicase, DNA polymerase, and ligase. So when we think of DNA helicase, we can see this as the thing that unzips the strand of DNA. So since DNA is double-stranded, you're going to need something to pull apart these double strands so that DNA polymerase can come in and start adding bases. So that's why DNA helicase is there. Now, DNA polymerase is basically going to be there to start adding the bases to these unwinded strands of DNA. And ligase is going to be the glue that puts it all back together. Now, building takes place in the 5' to 3' direction, and the difference between the leading and the lagging strand can be explained as the leading strand is replicated continuously, whereas the lagging strand is replicated discontinuously in short sections, and these short sections are what is referred to as Okazaki fragments, and they're called Okazaki fragments because they're essentially just shorter lengths of DNA. So whenever you're replicating the lagging strand, it's not going to be completely consistent and happening continually. You're going to have breakups in the process. So whenever we take a look at this picture, it basically just recaps everything that we talked about. Here we can see DNA polymerase helping add all the bases, and this is the lagging strand because, as you can see, the replication is not simultaneous like it is here, and helicase is unzipping the genes, and then at the end, ligase will come back and basically put all the seams back together. So now we're going to talk about the flow of genetic information from DNA to RNA to protein. Now, the thing that you should always remember through all your biology classes is the central dogma of biology. And basically, it's just that DNA goes to RNA, which the RNA will eventually be the one that makes the protein. So whenever DNA exists in the nucleus and mRNA or messenger RNA is formed, the messenger mRNA is ultimately what's going to be sent over to the ribosome so that a protein can be synthesized. So in the process of transcription, this is whenever we go from DNA to mRNA, and this happens in the nucleus, and then after the pre-mRNA is modified so that it's ready to pass on to the ribosome so that protein can be synthesized. This process is called translation, and the mRNA basically swims through it basically exits the nucleus through pores and then will eventually make its way onto the ribosome so that the protein can be synthesized. Now, genetic information can be seen as codes, and these are called codons. So these are eventually the things that are going to be translated into the amino acid sequences. So some differences between DNA and RNA are that RNA has an extra base pair, but it doesn't contain thymine. So in RNA, adenine is going to bind with uracil in place of thymine. So on a test or in a textbook, if you're ever given a sequence and you notice that there is a U in it, you know that you're looking at a strand of RNA, because in DNA, the uracil does not exist, whereas in DNA, you're just going to have adenine, guanine, cytosine, and thymine. So in both of these, we have the existence of triplet codes, and these are ultimately what codes for amino acids. So you guys are probably familiar with that lengthy chart that allows you to put the different triplet codes together so you can see which amino acid is being transcribed. This is the best representation of what the triplet codes are because these basically exist to code for an amino acid. So as we can see here, we have ATG, and then, or a better example is that AUG is a triplet code, and this will always code for the start codon, which is methionine. So now we're going to discuss how the genetic code dictates how codons are translated into amino acids. Now, In the genetic code, you're going to have those triplet codes, and then eventually these triplet codes will code for the amino acids that will make up the polypeptide chain, and these polypeptide chains are ultimately going to be what our proteins are. So there are 20 different amino acids that exist, and there are 64 triplets. Now the genetic code is universal because these bases can be seen in multiple different organisms. They're not limited to just humans or a specific type of animal. So these are the diagrams that I mentioned before, and basically these are what you use to decipher which amino acid is being talked about. So as we can see here, one code can code for more than one amino acid, and this is actually very helpful because say you have a mutation, it's not always going to be disastrous because, like in this example, if we switch a G for an A, we're not going to have any difference in what amino acid is being coded for. So the process of transcription can be broken down into 3 distinct parts. So in initiation, basically the DNA molecule is going to unwind and it will separate to form a small open complex. And then basically in elongation, the polymerase is going to move along the template strand and it will synthesize an mRNA molecule. Now here we're referring to RNA polymerase, not DNA polymerase, because we're synthesizing a strand of mRNA. And lastly, in termination, in prokaryotes, there are 2 ways in which transcription can be terminated, and they're all dependent on a Rho factor. So basically in Rho-dependent termination, the protein factor Rho is going to be responsible for disrupting the complex that involves the template strand, which is the RNA polymerase and the RNA molecule. But in Rho-independent termination, a loop will form at the end of the RNA molecule, which will cause it to detach itself. But termination in eukaryotes is more complicated because it involves the addition of more adenine nucleotides to the 3' end of the RNA transcript. So this is referred to as polyadenylation. Now in this picture, we basically just have an overview of what was just discussed. So here we can say in initiation, the transcription factors are binding to the promoter, and the promoter is located downstream from the actual start site, and here the transcription factors will bind just so the cell can get a sense of transcription is starting to occur. Now in elongation, the RNA polymerase is going to move along the DNA and synthesize the RNA transcript. And lastly, in termination, we can see that the termination signal is reached so that the RNA polymerase can disassociate from the DNA, and this can happen in the 3 different ways that were discussed on the last slide. So we're going to get into talking about how eukaryotic RNA is processed before leaving the nucleus as mRNA in part 2. So you guys can just stay tuned for the next video and leave any questions or comments that you had on this part in the comment section below, or you can email us at brhssciencehonors@gmail.com.